So we finished with, so what we figured out, one kind of a segue here, is that duodenal digestion depends on the activities, secretory activities of pancreas and liver, right? That makes sense? So that liver releases bile and pancreas releases a pancreatic juice, so-called pancreatic juice, which consists of bicarbonate and pancreatic enzymes, into the duodenum. Okay? Does anyone remember what we just talked about? Which hormones, two hormones that regulate activities of those two organs of the liver and the pancreas? Secretin and CCK, right? Okay. So, liver. Well, first of all, it tastes good. I don't know about human, but cow liver definitely tastes good. And it's the second largest organ in the human body. Largest internal, second largest overall. What's the largest? Skin, yes. So 2% of the body weight, so mine should be probably like 3.5 pounds. But of course, it's you know, it's not exact. If you would look at those four models right there, the guy on the left, next to the really dark brown model, the guy on the left, the liver is inferior to the diaphragm. It's right below the diaphragm, and it occupies most of the right upper quadrant. So, showing it on myself, it's going to be something like that. Okay. I should have worn this T-shirt again today. So hepatic artery and delivers the blood there, okay, to the liver tissue. Hepatic vein drains the blood away from the tissue. It's divided into 4 lobes. The large right lobe, you can see it here. The smaller left. So these two right and left lobes are the main, okay? They're separated by so-called falciform ligament, which runs in between, okay? Falciform ligament attaches the liver to the anterior wall of the abdominal cavity, right? The wall that is removed from the dude, actually, permanently removed, okay? Pretty much the entire liver is covered by the very thin layer of connective tissue that's really easy to penetrate. Okay, the only part of the liver that is not covered is the so-called bare area, where the liver attaches to the diaphragm. Actually, the top of the liver attaches to the diaphragm. The coronary ligaments suspended to the diaphragm, and we just mentioned 2 lobes, but I told you that there are 4. So there's caudati and quadrati lobe, okay? Small lobes. Okay, you can see that this is the caudati lobe of the liver, and this is quadrati lobe of the liver. Okay, so I have right, left, caudati, which is on top, and quadrati, which is on the bottom, and both smaller lobes are located on the posterior surface. Okay, a few more structures that I want to point your attention at regarding the liver. Well, the gallbladder that we're going to chat about, okay, here, hepatic artery and hepatic vein, okay, and this, you see that crevice? That crevice is the sulcus, so the deep area, okay, where the vena cava runs through. Okay, that's what that's about the anatomy. Now, the microanatomy of the liver gives really great insight into the relation between the function and the structure. So liver is often referred to as filter. Well, it's not only filter. It's chemical remediation plant. It's the filtering organ. It's the nutrient storage organ. It's the blood remediation plant, if you want. Liver has an enormous number of functions, as we will be able to see. Now, if we would look at the microanatomical structure of the liver, we will notice that liver consists of multiple hexagonal lobules. Okay? Each lobule is the functional unit of the liver. That makes sense? Now, let's take a look at this picture of the lobule and figure out what it consists of. First of all, liver consists of the special cells called hepatocytes. You can see them here. Okay? They're called hepatocytes because they perform a very specific function. Okay, they're differentiated cells that their function, the main function, is to produce bile. And when hepatocytes produce bile, this bile has to enter somewhere. Turns out it enters into those little tiny vessels. You see them, they're labeled green here. Okay, they're called bile canaliculi. So canaliculi is the fancy word, Latin word, for channel. Okay, so those vessels, they collect the bile that is produced by these hepatocytes and drain the bile into the bile ducts. Okay, see that? Now, hepatocytes are organized in plates, and between plates are the blood vessels. These blood vessels are called liver sinusoids. These are capillaries, and we didn't learn about different types of capillaries, but let me just tell you that sinusoids are the most permeable, okay? Extremely permeable. So when I say permeable, it means that there is a lot of exchange going on between the blood in the sinusoids and the hepatocytes. Now, in addition to the bile production, as I mentioned, what else does liver do? Huh? Nutrient conversion, okay? For instance, it will take in fructose and convert it into the glucose, okay? It will store glucose in the form of glycogen. If there is an excess of glucose, liver will facilitate the fat production and send it over to store somewhere, okay? It's responsible for cholesterol synthesis and excretion, hormonal excretion. So a lot of chemicals have to be shuffled between the blood in the sinusoids and hepatocytes. That makes sense. So blood flow, as you can imagine, in these blood vessels is really slow. It's not stagnant, but really, really slow, because you gotta have time for exchange. Does that make sense so far? Another structure that we're gonna find there, well, not structure, but types of the cell, you're not gonna see them on the picture. Reticular endothelial cells. They are also called Kupffer cells. And essentially, those are macrophages. Now, the blood, it goes through the liver, through those sinusoids. Where do you think it comes from in the first place? No. Yes, the intestines, right? Because liver has to convert the nutrients, so that blood comes from the intestines. What about the food that you eat? Is that clean? The food that you eat, is it clean? Not really. I mean, let's be honest. It's not, okay? Some of those microbes are being dealt with in the intestines. Some of them can make their way through the wall of intestine into the bloodstream and end up in the liver, right? And now macrophages in the liver are the ones that will catch them and destroy them. Does that make sense? They are actually located in the walls of the sinusoids. Does that make sense? Like little watchdogs. Okay? So far, yes. Reticular endothelial cells or macrophages. Okay? These two. Kupffer cells. I don't find particular amusement in forcing students to memorize a number of names. For instance, hepatopancreatic ampulla. Remember when liver, the bile duct, pancreatic duct, this guy. I mean, when I say hepatopancreatic ampulla, at least you have an idea. Okay, we talk about liver, pancreas. Well, it's probably to the duodenum. It has a name. I don't remember the name of the dude. Seriously. There's a dude that discovered it. And some people refer to it as ampulla of whoever. Why? Like, when I say cerebral aqueduct, do you know what I'm talking about? Cerebral aqueduct, CSF circulation. If I will tell you aqueduct of Sylvius, you probably wouldn't know that. But it's the same structure. So I try to avoid names as much as possible, names of people, because most of them, well, all of them are dead now. So we figured out the sinusoids, we figured out bile canaliculi, but we never spoke about the origin of the blood that goes through the sinusoids, right? And we never spoke about, well, we talked about where bile comes from, hepatocytes, right? But what about blood? Well, it turns out that blood goes from portal veins and arteries right here. Okay, so portal veins and arteries, blood in those, actually both arterial and venous blood enter the sinusoids. Okay, and sinusoids take it to the central vein. Okay, central veins collected into so-called interlobular veins, and interlobular veins then take it to the vena cava. That make sense? Think about the path again. So you have blood from the branches of hepatic artery, which are these essentially, the portal arteries. You have portal veins, the wrong arrow. You have portal veins that carry blood from the intestine. That mixed blood goes through the liver. Whatever is necessary is done to the blood. Glucose is taken in, blah, blah, blah. Fructose is taken in, converted, all that stuff, okay? And eventually this blood ends up in the central vein. So this hole in the center of the lobule is the central vein. Does that make sense? Now, central veins from many, many, many thousands and thousands and thousands of lobules collect the blood into the interlobar veins, and interlobar veins, or interlobular veins, all come together into the hepatic vein. And then hepatic vein end up in the vena cava. And vena cava brings the blood with all those nutrients to the heart. And then heart distributes that blood all over the body. Does that make sense? Now, the structures in the corners of the lobule, which consist of the portal artery, portal vein, and the bile duct, are called portal triads. Okay, they all are bunched together. Does that make sense? It's a portal triad. So we have here, think about this, we have two independent processes. I want you to think about this lobule and its function on the grandeur scale, okay? So I have two independent processes that happen in hepatocytes. One, bile production. Bile is produced by hepatocytes through the bile canaliculi, goes to the bile ducts, and then is collected and dumped into the duodenum. And the second parallel process is nutrients that are absorbed in the duodenum via the portal circulation go to the liver, mixed with the blood that delivered to the liver through the arteries, enter sinusoids. Whatever's necessary is done by same hepatocytes that produce bile. Okay, at the same time they convert sugars into fats and whatnot, and after that processing, blood rich in necessary nutrients. That's important. Not just whatever was taken from the intestine, but rich in necessary nutrients. That blood enters finally in the vena cava. Okay? So liver kind of in opposite directions, it produces bile that goes into the duodenum, and it processes the nutrients that go from the duodenum. That makes sense? Bile. Brownish, well, greenish brownish, watery fluid with the weakly alkaline pH. Contains bicarbonate ion to neutralize the acid. Bile salts, which derive from cholesterol molecules, and their function is to emulsify the lipids, and we're going to talk what the lipid emulsification is. And lecithin, another molecule that helps to emulsify the lipids. Bile pigments, which give it a color, not to make it pretty. There's nothing pretty in the bile, but to get rid of hemoglobin degradation products. Cholesterol, we're getting rid of cholesterol via bile. Mucin. Mucin is the component of what? Mucus, yeah. And mucus is sticky, so it helps you to keep food together. Yes?
So does the bile get put back into the duodenum, or does it get put like the jejunum or the ileum?
Oh, bile enters, great question. Bile enters specifically into the duodenum, only.
So the food comes from the stomach into the duodenum, and then the liver produces bile that goes back into the duodenum.
That goes, yes, so food that enters here, so say this is the bolus of food, okay? This bolus that just entered the duodenum will get mixed with the bile. Does that make sense? And that mixture will then proceed to the jejunum and to the ileum. Okay, did I answer? Because don't hesitate to tell me you answered different question. Okay, because sometimes it happens. Bile salts and bile acids. Well, it should say actually salts. Why I said bile acids? I didn't notice, you know, that's what, when you look at it like 1,000 times, you didn't notice this thing. So bile salts. They are derived from cholesterol. Okay, now this cholesterol, remember your endocrine system, cholesterol, is that fat soluble or water soluble molecule? Fat soluble, right? So this is fat soluble part. Now, on one end of that cholesterol molecule, if we can assign it an end, there's an amino acid residue, fat or water soluble? Water, okay? So that's water soluble. So you end up with so-called amphiphilic molecule that has lipophilic, fat soluble, and hydrophilic parts. Okay. Now these guys can form micelles, not two separate words, not my cells. The micelles. Micelle is a structure when you have something fatty inside. So the fatty parts, see the fatty parts, are facing inside of the micelle. And outside of the micelle, those little black sticks, okay, those are hydrophilic parts of the bile salts. Does that make sense? Inside. Now think about this. What is generally the environment? What is in your intestines? Water or oil? Well, on normal occasion, water, right? Unless you drank the entire bottle. In this case, I'm really sorry for you. So water. So those micelles have to exist in the watery environment. So they're going to have hydrophilic on the outside, but they're going to be hydrophobic on the inside. Does that make sense? And the function of those micelles, if you think about it, is to cover something that is not water but fat soluble. They give a droplet of fat some kind of a coat that's hydrophilic on the outside. That makes sense? So you have droplet of fat in the center, okay? Does that make sense? That black stuff is the droplet of fat. It freely interacts, it loves to interact with the non-polar parts. And the polar parts on the outside allow this micelle to normally exist in the watery environment. And you have such a thing, such a reagent in your kitchen that forms micelles. Allows you to solubilize fat. Any idea? It's not eatable. No. Unless you're a freak. What do you use when you need to wash a soap? Soap or dish fluid thing, liquid. It contains the fat soluble part and water soluble part. Okay? Now, you can do this experiment at home. It's very easy. If you take a little bit of oil and put it in like a skillet with water, oil's going to mostly float like several big blobs, like really significant blobs. You add the fluid, and they kind of immediately, those blobs are broken apart in a really tiny, smaller, like bubbles. Those are micelles. So you immediately solubilize fat. Okay? Does that make sense? You remove it and you pretty much raise it. If you wash the surface of the skillet, you raise the fat from the surface of the skillet and move it into the water. Does that make sense? That thing works exactly the same. Okay? It breaks down large blobs of fat. Well, relatively large. Not like these blobs. Not these blobs, but smaller blobs of fat into much smaller droplets. And we'll chat why it's important. Other components. The bilirubin. That's what gives bile a greenish color. When your red blood cells die, and they die after about 2 months, they release a lot of hemoglobin. And hemoglobin contains a lot of heme, which is an organic molecule containing iron. Iron's precious. Iron gets taken out of him. That sounds funny. Out of that organic porphyrin ring and is recycled. It's actually taken back into the bone marrow, yes. Absolutely, yes. And we need to get rid of it because it's toxic. We need to get rid of hemoglobin, okay? Because hemoglobin is fairly, well, globin is dispensable, the protein part. Heme part is also pretty easy to make, but iron is what the body cherishes. Iron is being conserved all the time, and it's really, iron regulation is really important. So heme is broken further into the carbon monoxide. Turns out we do produce carbon monoxide. And through the series of steps, okay, it is converted into the bilirubin. Does that make sense? Just, series of steps. So heme that is toxic is taken out of the blood and converted into the bilirubin. And bilirubin is excreted with the bile and goes into the intestines. And that's what gives feces the brownish color, the bilirubin. Okay? Now, some of the bilirubin in the intestines is converted into another molecule that is called urobilinogen. So we can say bilirubin converted into urobilinogen. Okay? Now, urobilinogen, when it's in the intestine, it can either leave the intestine with the feces, which is normal, or remember, intestines, there's always an absorption there, right? The absorptive process. So some of the urobilinogen gets absorbed into the blood, but we don't need it. So it's a chemical that has absolutely zero physiological meaning. So urobilinogen is then excreted through the kidneys with the urine. And that's what gives the urine, well, not brownish, but yellow color. Yeah. Does that make sense? Yes. I cannot tell you about the chemistry that's going on there. I have doubts that it has urea there. Probably urobilinogen name is derived because it goes into the urine and can be isolated from urine, but I doubt that it has anything to do with the urea. So chemically, it's a different story. But it's just, for me, it's just fascinating how the soul. So pretty much, your urine is yellow because of the heme degradation product goes through all these hoops to get absorbed in the small intestine, end up in the blood, and be excreted in kidneys. Okay, and by the way, this carbon monoxide that you produce, now there's some body of work that suggests that carbon monoxide is really important for the proper cardiac health. It's actually like anti-inflammatory and cardioprotective at the very, 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 very small levels that we make. It doesn't suggest that you have to kind of insert the exhaust pipe of the car and start breathing. Okay, so liver. It's a major excretory organ. Okay, so it, first of all, it excretes the bile salts. Okay, it's still excretion. Those bile salts go into the small intestine. Okay, now bile salts, since they play a very important function, they solubilize lipids, they get reabsorbed. You can see that illustration here. So this is the bile duct, okay? And bile enters the duodenum. Then some of the components of the bile are reabsorbed by the so-called hepatic portal circulation, the portal vein that takes nutrients from the duodenum to the liver. This circulation also picks up the bile salts. Right? Lecithin. Most of it is excreted. It's another molecule that's derived from cholesterol. It also helps to break down fat into smaller chunks. If you will read the composition of chocolate, one of the components of chocolate is lecithin. Not this one. No, it's not from human bile. No, it's actually from the plants. Okay, but the good chocolate, really good chocolate, if you read the composition, it contains a lot of cocoa butter. Now, that's butter, that's fat, and to avoid chunks of fat in your chocolate bar, the manufacturer adds lecithin so that butter becomes really, really, really fine and solubilized and spread through the chocolate. That makes sense? And bilirubin. Well, we talked about it. Bilirubin is the heme degradation product. Now this belongs to endogenous products, products that are produced in your own body. Bile salts are produced by the liver. Bilirubin is produced by the liver. Lecithin is produced by the liver, right? Okay. Exogenous drugs. A lot of drugs are excreted through the liver, okay? Now, why I pay so much attention to that? Because if you know whether the drug is reabsorbed right here or not, will determine what the dose of the drug should be prescribed. Think about this. If you prescribe something like ampicillin or tetracycline, it's a sort of a more modern drugs, okay? They are readily reabsorbed into the hepatic portal circulation. So imagine this. You have a drug, it circulated through the entire body, ended up in the liver, excreted in the intestine, but then most of it is taken back. So it can make another cycle, okay, and do stuff. So its concentration in the blood remains high. When you use some older drugs, like sulfa drugs, or penicillin, they will have almost no reabsorption, and most of them will get excreted with feces. That makes sense? So, if you take penicillin, you know that usually it's like 3-4 pills with a gigantic concentration a day. Some others, it can be 1 or 2 per day. That depends on the so-called pharmacodynamics of the drug, the changes of the drug level in the blood, and that will be partially determined by its excretion via the liver. Does that make sense? See that you've got microscopes. It's really tempting to play with them, but it's also really tempting to finish liver. You know what? We're going to pause like here.